the structure and function of the immune system. So আজকে আমাদের agenda হলো we will see how exactly the immune system is formed in the body. What are the organs involved? What are the cells involved? তাদের কি কাজ? Okay, and how do they function? Then we'll go into details of B cells, T cells, their development, their maturation, and then we'll read about something known as cytokines. Cytokines এর নাম শুনেছো? হ্যাঁ, কি করে cytokines রা? What are cytokines? Soluble? Yes. So what do they do actually? জোরে বলো। Lysis. Okay. So they help in the effector functions of the immune system. So we'll read about it. Okay, sit down. Now, if you see, the immune system is actually composed of some collective organs, the lympho-reticular system. That means there will be some lymphoid particles and there will be some reticuloendothelial cells involved. Now, there are two kinds of responses. One is a very specific response, okay? The response that is only harbored, the response that is only targeted to the antigen, or there are some responses which are non-specific. So, it will be there present. So, that specific response will be mounted by the lymphocytes and the plasma cells, very important. And non-specific response will be present for all. It will be targeted by mostly macrophages or the phagocytic cells. Now, immune response jodi bola hoy, then we have two kinds of immune response. One is a humoral response and another one is a cellular response. Humoral response ki? Humoral response is always antibody mediated. Antibody ko prakar? How many kinds of antibodies are there? Ki ache? Which is a secretory antibody? I see. Okay. Now, this is humoral is antibody mediated. All of you know antibody is secreted by which cell? Plasma cell. Correct? And the cellular immune response is caused by the sensitized lymphocytes. So, we will get into the details of it. Now, coming to the organs. এই যে এত plasma cell, এই যে এত lymphocytes, এই সব কিছু কোথায় থাকে? কোথা থেকে তারা জন্ম নেয়, কিভাবে কাজ করে? That is what you should be focusing for the next 30 minutes. Lymphoid organs এর মধ্যে কি আছে? We have central organs, that is the primary organs. কারা কারা? The bone marrow, and the main important organ is the thymus. Okay? Thymus কোথায় থাকে? You have read anatomy. Tell. Last bench brown shirt. বলো। You are the most visible person in the last bench. Rest of the people are hiding their heads behind the other person's head. Tell. Where is thymus located and how is it like how does it look, the organ? Thymus e kothay thake? এখানে থাকে বললে হবে না, এখানে এটা কোথায়? অ্যানাটমি তো পড়েছো, তো বলতে পারছো না কেন? আমি অ্যানাটমি পড়েছি 2005 সালে, তুমি অ্যানাটমি পড়েছো 2023 সালে, বলো কি? এটা না বলতে পারলে মুশকিল। কোথায় থাকে থাইমাস? বোঝো? হ্যাঁ, posterior mediastinum, কাছাকাছি গেছে, বোঝো? Okay, so the secondary organs which also play a very important role is spleen, okay, lymph nodes, MALT, and other lymphoid tissues in certain organs. So that will be covering it. Now, what is MALT? MALT. Jano? Ki? Mucosa-Associated Lymphoid Tissue. Correct. And the last one is the lymphoid cells, that is composed of the lymphocytes and the plasma cells. Now, thymus, as I have asked, it is located behind the upper sternum. There are two lobes and there is a fibrous capsule, and it is divided into an outer cortex and an inner medulla. Okay, these things you people will be knowing better because you have read in the last year. See, both this cortex and medulla will be housing these green cells. What are these green cells? Thymocytes. Okay. So there will be some immature or small thymocyte in the cortex, and there will be some mature thymocyte in the medullary region. And all of this region is divided into sections. These things that are called the trabecula. Okay. Now there are macrophages, there are blood vessels inside it, and there are something known as Hassel's corpuscle. Okay. So the thymus is divided broadly into the cortex and medulla. Cortex will be housing the small lymphocytes, and the medulla will be housing some epithelial cells and mature lymphocytes. Now what does the thymus do? The thymus primary function is to production and proliferation of the lymphocytes. যত lymphocytes of production করছে, only 2 to 5% of the lymphocytes will be leaving the thymus, and the rest of it will be destroyed. Okay. Now there is something known as thymic selections, because some lymphocytes, the T cells that are recognizing the MHC complexes, so a recognition, a maturation take bole thymic selection. We'll see later what it is. And there is something called the death of the T cells. See, the T cells that cannot recognize the MHC antigen, they react with its own self antigen. So they are destroyed in an autoimmune procedure. And the cells which recognize are called the conditioned cells. So those are the thymus dependent lymphocytes of the T cells that are liberated from the thymus. Now what happens if their thymus is having a problem? What kind of problem? Congenital defect, thymus to name, or there is a aplasia, or there is a hypogenesis of the thymus. Now that syndrome is called the Dye-George syndrome. Okay. Now Dye-George syndrome ki hocche, there is a deletion of 22q11.2 chromosome. So what happens is, the babies are born like this. They have an asymmetrical face while they are crying. They have some problem with the palate. They have a cleft palate, and they have small ears and hooded eyelids. So they have a problem with the immunity because they don't have thymus. Key problem hobe thymus na thakle. Yes, so there will be the deficiency of T lymphocytes. Okay. And there will be deficiency of cell mediated immunity. Obviously T lymphocyte nei to cell mediated kotheke thakbe. And they are able to tackle some extracellular infection via B cell, but intracellular infection jodi hoy, then it is fatal. Ebong ei case, because these things are mostly immunocompromised, you cannot give any live attenuated vaccine. In any kind of immunocompromised individual, live attenuated vaccine dewa jay na. Keno? Because if the vaccine is not killed, if it is live, then a full blown infection can occur, because tader immunity nei. Okay. So that's the point. Now the next central organ is our bone marrow. Okay. Now bone marrow, you know the locations of bone marrow. So all the lymphocytes, they origin in the bone marrow, but the T lymphocyte, they develop in the thymus, and the B lymphocyte, that's the main point of talking in the bone marrow, they origin and develop in the bone marrow. Okay. Now there is a selection process, and there is also a timeline where you can see that in the fetus, IgM and IgD secretion starts from the 20th week, but they receive the maternal IgG, the fetus receives it, but own IgG develops by 2 to 3 years. সুতরাং ফিটাসের যদি কোনো রকম symptoms কিংবা কোনো রকম disease diagnose করতে হয়, if you detect the IgG from the fetus, that is probably the maternal IgG cross transfer, because own IgG takes times to develop. So that is the thing. Coming to the secondary lymphoid organs, primary hoye geche thymus and bone marrow. Now the secondary lymphoid organs, most important is the lymph node. I'll not go into the details of the lymph node locations, you will be knowing it. Lymph nodes are located along the lymph vessels. Okay. Now they are again divided into cortex, follicle, medulla and paracortical area. Now ek ekta jayga theke ek ekta jinish kintu eder harbor korche. See, the cortex will be harboring the lymphocytes, the follicle will be harboring again the B lymphocytes, medulla will be harboring the B lymphocytes, plasma cells and macrophages, paracortical area again will be harboring the T lymphocytes and interdigitating cells. So this paracortical area is actually a thymus dependent area, because they will be harboring the T lymphocytes. Now, what is the function of the lymph nodes? They filter for the lymph. Because they have macrophages, they will assist in phagocytosis, and there will be some proliferation and circulation of the B cells and the T cells. As you can see that these areas are the areas that are responsible for harboring these cells. So, they have a proliferation and maturation part also. The next organ is the spleen. Spleen ke ki bola hai? It is the largest lymphoid organ. Yes. So, the spleen is again divided by trabecula. It forms a capsule, and there is again a thymus dependent area, that is the central arterial surroundings. Okay. See, spleen is called the graveyard for RBC. So, all the RBCs, they are destroyed in the spleen only. What happens if a patient has an abdominal injury, a blunt trauma in the abdomen? Suppose a road traffic accident. So, which is the most commonest organ that is affected in the upper abdomen? Jano? Spleen. And if you cannot detect the splenic injury, the patient will succumb to death very soon. Okay? Because it is a highly vascular, and it's very difficult to control the bleeding. So, instead of repairing it, some portions or some cases, you have to remove the spleen altogether. So, those patients are splenectomy patients. So, you remove the spleen from those patients. Now, splenectomy patients ko ki habe? Because spleen is again a lymphoid organ. So, you will be losing some immunity. Correct? So, that's why whenever you do a splenectomy patient, you give these vaccinations, because these patients will be susceptible to Streptococcus pneumoniae, to Neisseria meningitis, to Haemophilus influenzae, and other bacterial infections. Okay? So, you give the bacterial vaccines to the patients who have undergone splenectomy, because there will be some problem with the immunity of the patient, because you will be losing the largest lymphoid organ of the body. Understood? Okay. Now, coming to the Mucosa-Associated Lymphoid Tissues, or MALT. So, what are these? These are nothing but, see, there is mucosa in our alimentary canal, and in this alimentary canal mucosa, or the respiratory or the genitourinary mucosa, there are some portions which are called Peyer's patches. So, these are aggregates, and some are called the lymphoid follicles because they are isolated. Okay. Now, these Peyer's patches, if you can see here, yes, you see this is the ultrastructure of the intestinal cavity, the intestinal wall. You can see that this is our epithelial lining. Okay. These cells, this epithelial lining, you can see the microvilli is there, but you see these short midget cells here. So, they are M cells. Now, these M cells do not have this microvilli. Can you see? M cells are microvilli blunted. They are not like the other epithelial cells. Now, these M cells, they are the portal of entry of many bacteria to our body. Okay. So, that is why M cell is important. Later, when you read Salmonella, you will see Salmonella, how it enters our body through the M cells and the Peyer's patches. Okay. So, the bacteria or the microbe, they come to the M cell, and because they don't have a microvilli on their surface, very prominent, they enter the gut, and they will be entering in this area, that is the lamina propria. What does the lamina propria have? They have a lymphoid follicle, and from this lymphoid follicle, see, this is a Peyer's patch. So, that was my point of discussion. This is a MALT tissue. Okay. So, this is a Peyer's patch. From there, they will be infecting the body and carried over to the next cell and henceforth. So, we will not go into the details of the pathogenesis, but I just wanted you to locate the Peyer's patch. So, this is where Peyer's patch is located. Now, coming to the MALT cells, again, they are rich in collection of the lymphoid and phagocytic cells. The Ig produced are G, A, M and E, but primarily they will be producing A and G. Both B cell and T cell will be present, and as I said, they are the common mucosal or secretory system. They are present in the gut, respiratory and other genitourinary system, and total area, if you see, they have around, it is approximate 400 meters square of MALT tissue is there in our body. It is approximate data, it is not absolute value. And if you see it is gut associated, then it is called GALT, and if it is bronchus associated, then it is called BALT. But mostly in the exam you will be asked for the MALT only. Now coming to the M cells, I said it's a portal of entry for many organisms including Salmonella, Shigella, Vibrio, poliovirus, etc. They'll be having T cell, B cells and macrophages, and B lymphocytes in the MALT, once activated at the site of antigenic exposure, they will be migrating to other parts of the body and they will be producing the local immunity or the secretory IgA at that position, okay, because B cells are there in the M cell. You can see here, see this M cell is actually a portal of entry for all these microorganisms, and from there, these are the plasma cells in the lymphoid follicles. From these plasma cells, the antibodies are produced, and those antibodies once activated, the cells migrate to other parts of the body and secrete and give the secretory IgAs to the local immunity. Now coming to the cells of the lymphoreticular system, we have talked about the organs, now we will concentrate on the cells. So what cells we have in a system? So we have lymphocytes. All of you know that lymphocytes are there in the blood, and this is a picture of the lymphocyte. You can see here, this is a lymphocyte, okay. So 20 to 40% of the total RBC, total WBC are the lymphocytes. 99% of them are in the lymph, and these are the three types of main lymphocyte: B cell, T cell and the natural killer cell or the NK cell. Now, if you see, some are short-lived and some are long-lived. The ones which are long-lived are the memory cells, and the ones which are short-lived are the effector cells mostly. There are some lymphocytes will be recirculating in the system, so mostly they are the T cells. This is the difference between three types of cells, that is the naive cell, that means the cells which are not exposed to the antigen or which are not mature, and then we have effector cells, and then we have memory cells. Okay, now if you concentrate on the first part of the chart here, see the location of these cells. First of all, the naive cells are present in the secondary lymphoid organs, lymph node and spleen. Those are the naive cells. Effector cells are located in the inflamed tissue and the mucosal surfaces. And the memory cells are located in both these areas, because some portion of the long-lived cell will be the memory cells. So they will be present in both the presence of the naive cell and the location of the effector cell. Okay. Now the morphology, these are the small lymphocytes, effector cell are mostly large lymphocytes, and memory cells are mostly the small lymphocytes. Lifespan, naive cells are short, effector cells are short, and memory cells are long, as I said. Now what is the function of the naive cell? The naive cell functions because they transform to effector cell on the primary exposure to antigen. So I told this, all of you understand the meaning of naive. সবাই naive মানে বোঝো তো, যে অতটা কিছু জানে না, new to the system, okay, those people or those kind of things are called naive. So naive cells মানে they are new to the system, they have not been exposed to the antigen yet, okay. So the cell proliferation has not been specified, so those cells are called the naive cells. Once they are exposed to the antigen, affected to some kind of specific response, so that is called the effector cell. Now what is the function of the effector cell? The function of the effector cell is to eliminate the antigen. আমাদের বডি কি চাইবে? একটা আমার অ্যান্টিজেন ঢুকলো, I want to kill the antigen, that's all. সেই অ্যান্টিজেনকে কিল করার জন্য যা যা সেলুলার মেকানিজম দরকার, যা যা সলিউবল প্রোডাক্টস দরকার, সেগুলো বডি will try to manufacture and supply. So those manufacture and supply is in the process of producing lymphocytes, producing B cell, producing T cell, producing cytokines, all these things, okay. Now the functions of yes, if you can see, T cell, B cell is involved in immune response. I told naive cells only the mature T cell and B cell before encountering the antigen. And upon reaction to the antigen, what happens is either you get the tolerance or you initiate the immune response. Tolerance kano? If you give a small antigenic exposure to an individual repeatedly, what will happen? You produce a tolerance to that antigen, so that the immune response is not yet mounted, and there is no effect on the body on the antigen again. Okay. Now T cells, what do? They produce the cell mediated immunity by cytotoxic T cells. So those are the effector organs, the effector organelles. B cells, what do? They produce antibody. Simple. Now surface marker, this is very, very important. In your whole microbiology, you will see certain things called CD antigen. Okay, CD antigen kothay thake? Thymocytes, they harbor CD1. T cell receptors, they harbor CD2. T cell antigen receptor complex, they will be harboring CD3. CD4 will be with the helper T cells, that is the T8 cell. T regulatory cells will be harboring CD8, and CD19 will be on the B cell. Okay. So the CD antigen will be associated with these kind of specific T cell or B cell. Okay. Now property of the T cell and B cell, I have tabulated it. You can read it when you go home. There's nothing to again explain in this thing. Now coming to the development of T cell, this is very important and it is there in your book also. Okay, eta boyer thekei newa. Now you see here in the bone marrow there is a hematopoietic precursor. Bone marrow theke kothay elo? The thymic cortex. Thymic cortex theke the T cell precursor develops from the hematopoietic precursor. Shekhan theke the double negative T cell, that is CD4 negative, CD8 negative T cell develops. তাহলে কি হলো? T cell precursor থেকে double negative T cell, সেখান থেকে কি হলো? When the T cell receptor complex, they develop either a gamma delta T cell or they develop a double positive T cell, that is the CD4 CD8 antigen positive T cell. সেখান থেকে a positive selection হলো 5%, and then again they develop MHC restriction molecule on it. Whenever there is a MHC restriction molecule on it, they further come into the thymus medulla. From the thymus medulla, they differentiate into the CD4 cell and the CD8 cell. Okay, CD4 cell, CD8 cell a differentiation holo, then that CD4 and CD8 cell will be in the peripheral blood tissues, sekhane they will be into the effector cell and the effector response. Okay, so that is how exactly a hematopoietic stem cell precursor is actually converted into a effector CD8 or CD4 cell for the immune response. Okay, this is a very important chart. Now T cell maturation ami bollam. One important thing is ei je T cell development hocche, association hocche, this all is controlled with the T cell receptor. So this is the ultrastructure of the receptor. If you can see here, there are two the parts of the receptor. There the two parts are connected by this bond. This is the disulfide bond here. You can see the disulfide bond at the base, and they have a short cytoplasmic tail here. Okay, and the transmembrane part, this part is the transmembrane part, this part is mostly the positive charged, and if you see upwards there are two pairs of glycoprotein chain. So these are the glycoprotein chain. One is the beta chain, one is the alpha chain. Okay, so ekdike alpha chain ache, ekdike beta chain ache. Transmembrane part ta positively charged, they have a short cytoplasmic tail, and this alpha and beta chains are connected by the disulfide bond. So this is the ultrastructure of the T cell receptor. They may give a question with the T cell receptor also, the structure and the functions of the T cell receptor and what are the antigens or the CD antigens that are associated with it. Now ei je bolchi MHC restriction, so the T cell will develop some MHC restriction because CD8, you can see here, CD8 will only respond to the antigen presented by the HLA class 1, and CD4 will be only responding to the antigen presented by the HLA class 2. If the HLA class 2 antigen is in contact with some cells with the CD8, they will not respond because they are not designed to recognize that molecule. Now what happens is mature CD4 plus the T cell receptor, as I described, will be inducing the helper or the inducer cells. তারা কি করছে? B cell differentiation and stimulation of the CD8 cells. Mature CD8 cell, if they come in contact with the T cell receptor, they will give rise to something known as cytotoxic T cells. Now what is the function of cytotoxic T cells? তারা কি করছে? Elimination of antigen by perforins and granzyme mediated lysis or the cytotoxicity. So the CD4 T cell actually helps in the B cell differentiation. The CD8 T cell is actually a cytotoxic T cell which eliminates the antigen or which eliminates the foreign particle by perforins and some enzyme called granzymes. Okay. Now this is again differentiation of the T cell. If you see the thymocyte here, thymocyte টা আমাদের মেন ছিল থাইমাসে, সেখান থেকে কি হলো? আলফা বিটা টি সেল হলো, আলফা বিটা টি সেল থেকে একটা ক্লাস্টার গেল CD4 T cell, একটা ক্লাস্টার গেল CD8 T cell. Now the CD8 T cell again matures and develops into cytotoxic T cell, and CD4 T cell থেকে কি হচ্ছে দেখো, there are many kinds of associated T helper cells that are being developed. কি কি হচ্ছে? T regulatory cells হচ্ছে, যারা কিনা the immune system টাকে regulate করছে, and there are certain development in the TH17, TH2 and TH1. Now this TH2 they activate the B cell, the TH17 they promote the inflammation, and the TH1 they are associated with the secretion of the cytokines. Okay, so this is very important. যে কোন সেলটা আমার CD8 T cell is targeting the antigen, they're trying to kill the antigen or the foreign cells with the granzymes and the perforins, and the rest of the CD4 cells are more concerned with the production and proliferation of B cells and the production of the cytokines and regulation of the immune system. So this is the whole motto. এই আর্মটা যাচ্ছে আমার সেল কিলিং এ, এই আর্মটা যাচ্ছে বি সেল ডিফারেন্সিয়েশন, ইমিউন রেসপন্সটাকে প্রলিফারেট করা এবং সাইটোকাইন প্রোডাকশন করা। ওকে, now this already I have told. আচ্ছা, মেমোরি টি সেল কিন্তু CD4 CD8 দুটো থেকেই হয়, so there is no problem in it. Now B cell maturation is a little bit more easy to understand. You can see there one is the stem cell, from there the pro B cell is developed. From the pro B cell there is a pre B cell that is developed. If you can see, this pre B cell has some molecule that is attached over the surface. What is this molecule? এই স্ট্রাকচারটাকে দেখে কি মনে হচ্ছে? Y স্ট্রাকচার, হ্যাঁ, antibody. Okay, now from the pre B cell the immature B cell develops. Okay, now the immature B cell, you see the structure is again matured, the structure is again developed, so they have something which is attached to the surface. We'll see what it is. From the immature B cell the naive B cell develop, from the naive B cell the mature B cell develop. এই mature B cell টা কি হলো না, they either go into development from to the memory cell, or they go into development for the plasma cell. Now this plasma cell will be starting to secrete the antibodies. So this is how a B cell develops and secretes antibodies via the plasma cell. একটা পোর্শন প্লাজমা সেল হয়ে অ্যান্টিবডি সিক্রিট করবে, আর একটা পোর্শন উইল বি ডেভেলপিং টু দ্য মেমোরি সেলস। মেমোরি সেল হয়ে কি হবে? হোয়াট ইজ দ্য ফাংশন অফ মেমোরি সেল? ফিউচারে কি হবে? Yes, to mount a rapid immunological response you need to have a immunological memory, and that immunological memory is imparted by the memory cells. Okay, T cell or B cell difference, this is very easy, but only thing that you have to remember is the markers. You see the CD markers in the T cell it is 3, 4, 8, and in the B cell it is 19, 20 and 24, 19, 21 and 24. You have to remember the CD markers. And the maturation is in the thymus for the T cell, in the bone marrow for the B cell, and all of them origin from the bone marrow itself. Okay, plasma cell যেটা বললাম ultimately the last stage of development of the mature B cell. They are antibody secreting cell or IG producing factory bola hoy. They are kind of oval. They have a eccentric location of the nuclei, and they also have the cell organelles like ribosome, endoplasmic reticulum, mitochondria, and the Golgi apparatus. They have a lifespan of 2 to 3 days, and once the plasma cell is produced, they can only form antibody of a single class. এই না যে একটা প্লাজমা সেল অনেকগুলো ক্লাসের অ্যান্টিবডি প্রোডাকশন শুরু করে দেবে, ওকে? They are designed in such a way, and they will be allotype of a single light chain production করবে, ওকে? অ্যান্টিবডি ক্লাস হচ্ছে তো, অ্যান্টিবডি হয়নি তোমাদের? অ্যান্টিবডি হলে এটা আরো একটু ভালো করে বুঝতে পারতে অ্যাকচুয়ালি। Okay, so the memory cells are expressed in all the isotypes of the antibodies, G, A, M, E, D. Now there is something known as B1 cell. B1 cell ta ki? There will be some production of the B1 cells from the fetal liver, okay? And these B1 cells are T cell independent, okay? So hence they produce some immunity to the neonate, okay? So that's a good point. Now coming to the next effector cell, those are the null cells. Null cells keno bola hoy? Because they lack a feature of B cell, and they also lack a feature of the T cell, but they are cells of the lymphoid system, they are cells of the immune response. So tara ki korbe? Taderke bola hoy large granular lymphocytes, okay? So they are double the size of the small lymphocytes, and they have some granulation, they have some abundant cytoplasm, azirophilic granules, they have endoplasmic reticulum, Golgi apparatus, and so on. Null cells are collectively called natural killer cells. They are also part of the antibody dependent cellular cytotoxicity cells, and they are also a part of the lymphokine activated killer cells or the LAC cells, okay? Now NK cells jodi dekho, NK cells have a lot of function in the terminal portion of the immune response. So what do? They are spontaneously cytotoxic. They don't need sensitization. সে যেরকম অ্যান্টিজেনই হোক না কেন, if they are produced and if they are targeted, they will go and kill the cell. They don't need sensitization, they are not antibody dependent, and they target the virus infected cell or the malignant cell, natural killer cell. They form a part of the innate immunity. Innate immunity পড়েছো তো? They form a part of the innate immunity, and the most important thing is they are still active in these cases of severe combined immunodeficiency where the B cell and T cells are absent. So they are the last hope for the immunocompromised patient, SCID patients, severe combined immunodeficiency patients. They all have the natural killer cell, but that doesn't mean natural killer cell will give you the immune or the immunity to all the viruses and all the bacteria in the world. No, they are present and they function in the SCID patients. They express certain CD antigens on the surface, CD16 and 56. And what is the effect? The effect is perforin or TNF mediated apoptosis. TNF mane ki? Tumor necrosis factor. Okay. ADCC is a similar mechanism where you can see these NK cells are harbored with some FC receptors on their surface. So whenever these kind of antigens are present on a cell, automatically this will be the perfect combination for the natural killer cell, and they will be activated, and then they will be producing the apoptosis. So these are the antibody dependent cytotoxic cells, ADCC cells, okay? They have a surface protein, as I said, a fraction of the IgG antibody is produced on the surface. So যেখানে ওরা দেখলো যে IgG coated একটা সেল আছে, সারফেস কোটেড যে আইজিজিটা আছে পার্শিয়াল, সেটা গিয়ে ওর সাথে বাইন্ড করে গেল, and from that binding they formally are transferred under the NK cells and they produce the apoptosis. So this is called antibody dependent cytotoxicity, ADCC. আচ্ছা, lac cells are কি? Lac cells, the NK cells treated with interleukin 2, they are from the lac cells. They are cytotoxic specifically for the tumor cells. They don't have a much role to the infection of the foreign antigens. Tumor cell recognize করে। আচ্ছা, phagocytic cells এর মধ্যে most important is the macrophage. It is the largest lymphoid cell in the peripheral blood. অনেক জায়গাকার ম্যাক্রোফাজের এক একটা নাম, অ্যালভিওলা ম্যাক্রোফাজ আছে, হিস্টোসাইটস আছে, কুফার সেলস আছে, অস্টিওক্লাস্টস আছে, মেসেঞ্জিয়াল সেলস আছে। There are many kinds of macrophages which are named individually to the each organ system. What do? They phagocytose the cell. You have read in physiology, পড়েছো তো? পড়নি ম্যাক্রোফাজ? হ্যাঁ। So they trap the antigen, provide it to the lymphocyte. কি বলা হয় জিনিসটাকে? Antigen presentation. You trap the antigen, you produce it to the lymphocyte. মানে হচ্ছে দারোগার মতো ধরে নিয়ে জেলে পুরে দিল, তারপরে জেলে সে পিটানি খাচ্ছে। ওকে, so if the macrophage is bearing a different MHC antigen, the T cell does not respond because ওই যে বলেছিলাম a particular MHC class antigen will be responding to a particular CD differentiating antigen only. Okay, T cell will be only accepting the processed antigen presenting by the self MHC. So there is definitely a parity between antigen and the MHC class that is presenting on it. Okay. Activated macrophage, they are the the efficiency of normal macrophages. They are increased by lymphokine, complement, and interferon. They are not antigen specific, and once they are activated, they are cytotoxic to the tumor cells and other organisms. So this is a variant of macrophage which is activated by several soluble products like cytokines. And what are their function? Very important. The function is to produce the hydrolytic enzymes. Hydrolytic enzyme, binding protein, TNF, colony stimulating factor, and interleukin 1. So these are the functions of the activated macrophages. Now what is a microphage? I bet you have not heard this name, microphage. Macrophage to sabai শুনেছো, microphage you have not heard. So microphage is actually collection of this polymorphonuclear cells of the blood. So those are collection of neutrophil, eosinophil, and basophil. Neutrophil কি করছে? They are actively phagocytic. Eosinophil কি করছে? They are producing the allergic inflammation. They are mounting a response for the parasite, and they are also mounting response for the antigen antibody complex. Okay. Basophil found in the blood and the tissues mostly as the mast cell. Basophil will be producing heparin, histamine, and serotonin. So all these allergic responses, you can see the activity of basophil. Now the next important thing is the antigen presenting cells. That means the cells which are capturing the antigen and they are presenting to the lymphocytes. Ki ache? Dendritic cell, macrophages as I said, and B lymphocyte. Macrophage discussion আমরা করে ফেলেছি. We are left with the dendritic cells. Dendritic cells are the major antigen presenting cell. You can see the part of the dendritic cell. Why they are called the dendritic cell? They are the professional antigen presenting cell. Okay. Now whenever an antigen has come inside the body, first the cell that will be encountering the antigen will be the dendritic cell. Dendritic cell will recognize the antigen. They will take the antigen up, they will present it to the lymphoid cells so that further effector system can be carried out. Okay. Now they differentiate, they are different from the B or T cell lineage in the bone marrow. So প্রথম থেকেই ওরা আলাদা হয়ে গেল। MHC class 2 is expressed along with the co-stimulatory molecules B7 and CD28. These B7 and CD28 are required for the TH activation. Okay. They are pleomorphic, they are small, having a central body, and they have a long needle-like process. And this long needle-like process actually helps them to capture the foreign particle of the antigen. So that's why they are called the dendrites. They present the antigen to the T cells during the primary immune response. Similarly, arekta type of cell called the Langerhans cell, they are also the APCs, but these APCs are located on the skin. So whenever there is a foreign particle or antigen on the skin, these Langerhans cells will take part. Okay. Otherwise, the dendritic cells, they are good enough for the whole system. Now MHC. MHC মানে কি? Yes. Histocompatibility বলা হয় কেন জানো? Why it is compatible? মানে এই টার্মটা এলো কোথা থেকে? They are mostly the major antigens or the major determinants for the transplant. তাই জন্যই compatible কথাটা এসছে, major histocompatible complex. So তিনটে ক্লাস অফ প্রোটিন আছে, MHC1, MHC2, MHC3. MHC1 কি করছে? They are associated with the receptance or the rejection of the allografts. MHC class 2, they regulate the immune response. MHC class 3, they induce the complement system. So their MHC classes have complete different functions. দুটো তিনটের মধ্যে কোনো similarity নেই। আচ্ছা, yes, MHC complex and HLA complex are synonymous. Okay, so this one point you have to notice. MHC is synonymous with human leukocyte antigen. So don't get confused, HLA ta ki holo or MHC ta ki holo, they are synonymous. I know the synonymous words. Now coming to the class 1 MHC, I'll not go into the details of the ultrastructure, but একটা জিনিস জেনে রাখা উচিত যে class 1 MHC are the glycoproteins which are harboring alpha chain and the beta chain. See, the beta chain is a very less amount and the alpha chain is the majority. Okay, so this alpha chain is a transmembrane glycoprotein and beta chain is only extracellular. Okay, beta chain টা শুধু বাইরে আছে, বাকি alpha chain টা is a transmembrane protein. MHC class 2, if you see here, is a heterodimer of alpha and the beta chain. So they have an equivalent proportion of alpha and the beta chain in their structure. MHC class 3, it induces the complement system. It is again a heterogeneous molecule. Now MHC টা আমি ডিটেক্ট করব কি করে? You see, before the transplant we generally do a HLA typing. HLA typing টা করা হয় কি করে? A mixed leukocyte reaction or primed lymphocyte typing. MLR or PLT short form বলা হয়. So these are the two methods that are used for the HLA typing. Application কি, as I told, transplant accept হবে না transplant reject হবে, সেটা একটা indication হচ্ছে HLA match. You have to do HLA match before a transplant. There are other things you can do. You can do a paternity testing, you can do some anthropological studies or some genetic studies. Now the last portion of the class, cytokines. The soluble products are called the cytokines. Okay, cytokines কি হচ্ছে দেখো, these are the chemical messengers. কাকে করছে? The interaction and communication between various cells of the lymphoid system. For example, they are called lymphokines. কারা প্রডিউস করছে? lymphocytes. They are called monokines. কারা প্রডিউস করছে? monocyte. Some are called interleukins, produced by the WBC. Some are chemokines, which are involved in the process called chemotaxis. Properties কি বলছে? They are comparable with the properties of a hormone. That means there will be some cytokines will be having autocrine action, some cytokines will be having a paracrine action, that is the nearby, and some cytokine will be having a endocrine action. Endocrine মানে কি? Distant, যেখান থেকে রিলিজ হলো অন্য কোথাও গিয়ে কাজ করবে। ওকে, প্লিওট্রফি and redundancy, these are the properties of cytokines. Pleiotrophy কি বলছে? The same cytokine will be having different actions on different target cells, that is the pleiotrophy. Redundancy কি বলছে? Different cytokines will be producing the same effect on the same target cell. Okay, so these are the properties of cytokines, nothing much. Acha, structure of cytokines, I will not go into the details, but they belong to either hematopoietin, interferon, chemokine or the TNF family. This is the classification of cytokines. Acha, function এর মধ্যে দেখো, there is a chart. I don't think the chart is there in this slide, but there is a chart in your book. You will be having antiviral activity, anti-tumor activity, pyrogenic activity, and some hematopoiesis regulation and some inflammatory response. The most important cytokines which are responsible for inflammatory response are IL-1, IL-8 and TNF alpha. So যেখানে inflammation, you can either find interleukin 1, you can either find interleukin 8 or TNF alpha. You have to remember these three. Okay, some diseases Some bacteria, they produce some organism like E. coli, Neisseria meningitidis, they produce some endotoxins. Now those endotoxins will stimulate the macrophage and they will be producing the interleukin 1 and TNF alpha and will be giving rise to the inflammation. Okay, and that inflammation will give rise to the septic shock to the patient. এই যে পেশেন্টের ব্যাকটেরিয়াল ইনফেকশনে সেপটিক শকে চলে যায়, the shock is mediated by the cytokines. Okay, there is another thing called toxic shock syndrome. These are produced by the Staphylococcus aureus. Systemic হলে তখন তুমি বুঝতে পারবে জিনিসগুলো. There is something called cytokine storm. মানে কি হচ্ছে, যেখানেই cytokines are produced in excess that is not tolerated by the system, that give rise to some problem in our body, is called a cytokine storm. কোথায় কোথায় হচ্ছে? Graft versus host disease, GVHD, in the transplant patients, then in the ARDS in influenza patient, in COVID-19, in sepsis, in Ebola, smallpox, SARS. There are so many places where the cytokine production is excessive, which the body cannot handle, and they produce some deleterious effect in their system. So that is called a cytokine storm. Some cytokines are good: interferon alpha, interferon beta, and interferon gamma. Hepatitis treatment hoy interferon alpha diye. So if you have a hepatitis patient, then you can give interferon alpha for the treatment. So that's a good cytokine.